So thank you very much to the organisers for inviting me to speak. It's a great honour to be part of this meeting. I'm going to talk a little bit about our work looking at polygenic scores, and in particular trying to address the question of why polygenic scores do not transfer across ancestries. So I imagine most people are familiar with the idea, but just so we're all on the same page, this is the Genome-wide Association study for height in UK Biobank. Now I'm going to talk a lot about height, not because it's necessarily the most important trait in the world, although it is very interesting in its own right, but really because it's a model for other complex traits. It's very easy to study, it's very easy to collect large samples, and we know a lot about the genetic basis of height, and in fact, many of the complex traits and diseases that we're interested in have similar genetic architectures, at least to the extent that we can tell. Now, as I said, we can conduct very large genome-wide association studies of height, and in the UK Biobank, there are somewhere around 3,000 independent genome-wide significant loci. But of course, the effect size at each one of these loci is tiny, on the order of plus or minus 1 to 2 millimeters. So knowing your genotype at any one of these loci doesn't really tell you very much about your phenotype. But we can try and account for this in perhaps the most naive way possible, just by summing up the effect of all of these variants. So we construct what we call a polygenic score by summing up the effect size beta multiplied by your genotype at each one of these variants. That gives you a single number, which is a prediction of your genetic value for that phenotype. Now, much of the subtlety in this construction comes from how we actually choose the SNPs to include or how we choose the weights. I'm not really going to talk about that in this talk. Most of what I'll say is actually independent of that. Now, of course, knowing your polygenic score for height might not be very interesting because, of course, you know your actual height. But a lot of the interest in this field comes from the idea that we might be able to use this technology for disease traits. And in particular, for example, here, Kera et al. Showed that if you stratify people by their polygenic risk score, you can identify people at very high risk of disease. So here on the x-axis, people have been binned by their percentile of polygenic risk for coronary artery disease. And you can see if you're unfortunate enough to be in the top percentile of risk here, you have something like a 5-fold increased risk of disease compared to someone around the median here. So that's very exciting because in principle, you could identify such people before they develop disease and then put them on some interventional course that might reduce their risk in the future. So that's where a lot of the interest comes from in polygenic scores. My lab is particularly interested in the idea that we could use this technology to actually track the evolution of complex traits over time. So what I'm showing you here is the change in height polygenic score over the past 38,000 years in Europe. So each point here is an ancient individual from whose genome has been sequenced, or at least has genome-wide data. And we find these significant mean shifts in polygenic score, for example, one here going from the early to late Upper Paleolithic. This is where the Ice Age happens, and two going from the Neolithic into the Bronze Age here. Okay. And what we show in this paper is that these changes in polygenic score actually track changes in actual height measured directly from skeletons. So we show that we can actually use polygenic scores, at least in this case, to track the evolution of complex traits over time. In fact, we can use polygenic scores also to learn about individual ancient properties as well. Here we show with about 150 ancient Europeans from whom we have both genome-wide data and skeletal measurements. These are people who lived and died sometime in the past 10,000 years, that we can explain about 8% of the phenotypic variance in those individuals. Now, given that these are all low coverage data, that's about what you would expect. The maximum coverage here is about half of what you would get in a present day sample. So in fact, we can use polygenic scores, at least again in this case, to study complex traits, not just in living people, but also in ancient people. So that's all very exciting. But of course, there are problems with polygenic scores. One of the problems that's been heavily discussed is that they don't transfer very well across ancestries. Here in a paper by Alicia Martin and colleagues, they show that if you conduct GWAS in a European ancestry population, and you then go and try and predict phenotypes and populations of non-European ancestry, you lose a substantial amount of predictive power. For example, about half in East Asian populations and about 3/4 in African ancestry populations, specifically African Americans. But actually, an even more worrying problem is that sometimes polygenic scores don't even transfer within ancestries. And in this somewhat alarming paper by Mostafavi et al., they show that if you conduct a GWAS in a sub-cohort of UK Biobank, and then try and predict in another sub-cohort, and they stratify those cohorts in different ways, you can actually lose a substantial amount of predictive power. So there's no difference in genetic ancestry here. This is really saying that there must be environmental or other interactions. Now, obviously, both of these effects are quite alarming if we're trying to actually use these polygenic scores in a clinical setting, diverse patient populations, or indeed if we're trying to use them to learn about the evolution of complex traits in different populations. So that brings me on to the question of my talk, which is really, why do polygenic scores not transfer in these ways? What's driving these limitations? Now, one reason is, of course, differences in LD structure and allele frequency across populations. This has been heavily discussed elsewhere. I'm actually not going to talk so much about this today, although it is certainly a major component of this issue. But what I'm going to talk about are two other issues which I think are also very important. The first is the question of population stratification in the discovery population. This is work by Arslan Zaidi, who's a postdoc in the lab. And I think most people in the GWAS field will be familiar with the idea of population stratification as this problem, which really just keeps coming back and back over and over again. So what Arslan did was to set out to try and understand how relatively low levels of population stratification could affect polygenic scores. So we tried to simulate a population that was in many ways similar to, say, UK Biobank. So we simulate a structured population here in a 6x6 grid. So each population lives in one of these demes. And there's some environmental structure. So if you live in the north, you have a higher phenotype, but for purely environmental reasons, there's no genetic variation in the phenotype. All the genetic variation is neutral. And the demographic history of these demes is given by this model. So the demes are separate and there's some migration between them here. And then about 100 generations in the past, we collapse all the demes into a single panmictic population. And that's inspired by what we know about the time depth of structure in Britain based, for example, on ancient DNA. We choose these migration rates to be relatively high so that the structure in this population, again, is similar to what we observe in UK Biobank. So that's an FST of something on the order of 10 to the minus 3. So really very low level of population structure. So when we run the standard GWAS in this framework, this is the QQ plot. So on the x-axis, the expected log 10 p-values, and on the y-axis, the observed log 10 p-values. And what you see is that there's some inflation, at least in the common variance, shown by the blue line being above the expected y equals x line. That's, of course, what we expect to see when we do association studies in a structured population. But there's not a lot of inflation. Okay. So in particular, we're not reaching the levels at which we would expect to see genome-wide significant false positive associations. And so that's consistent with our sort of intuition that the amount of structure that we see in a population like UK Biobank is not enough to cause genome-wide significant false positive hits. In that sense, we're relatively safe. But what about when we look at the PRS? Then we see something quite different. So if we actually know the causal variance and just build the PRS out of the causal variance, then we don't see any structure. These are maps of the relative value of the PRS corresponding to the map of the environmental structure. Okay, and of course, we know that that's the case because we simulated that there is no true genetic structure. But of course, in practice, we don't know the causal variance. We have to try and infer them from the GWAS. And if we do that, for example, using the lead SNPs, as is often done, we find that the PRS is actually stratified. Now, we know this is spurious because we know there's no true genetic difference in the PRS. So we actually, what this tells us is the PRS is actually recapitulating the environmental structure. Okay, so some of the predictive power of the PRS is effectively coming from the environment. If we try and correct for this using principal components, it doesn't really make any difference, at least when we compute the principal components from common variants. That's because really this demographic history is too recent to be captured by common variants. If we use rare variants in this case, then it actually does correct for the population structure, and that's because we can capture this fine scale population structure. Of course, the problem with this is that in general, we don't have rare variants available from genome-wide association studies. We've use genotyping arrays which only really capture common variants. So that's one problem, but another problem is that actually if the environmental structure perhaps more realistically is not smooth with respect to any of the principal components, then actually even that doesn't work. So here we've simulated a case where a single deme actually has an inflated phenotype, and again, although we're correcting for the genome-wide false positive associations in the GWAS, that the polygenic score is actually still showing stratification. So in some sense, I think what this means is that we simply can't really correct for population structure in the polygenic score. In some sense, the polygenic scores are much more susceptible to population structure than actually even the original GWAS. Those were sort of somewhat artificial simulations, but just to make the point, we actually did simulate what we think the UK Biobank is like. So this is the best sort of demographic model of the history of Britain that we could come up with based on ancient DNA, and we simulated the actual geographic structure and ascertainment scheme of the UK Biobank, and we find much the same results. If we use common principal components to correct for population structure, we see this environmental gradient, and then if we see and then when we use rare principal components, we can correct for population structure, but actually not in this case of sharp structure. So that's the first part of the talk, looking at population stratification. And really the takeaway there is that we actually can't really control the extent of the effect of population structure on the polygenic score. Now, the second part of the talk really focuses on a different question, this question of whether there are differences in effect size between populations. And this is work by Barbara Bittarello, who until recently was a postdoc in the lab, has now started her own lab at Bryn Mawr College. And the way Barbara studied this was by looking in admixed populations. Okay. In particular, populations of admixed European and African ancestry, so that would be African Americans, and also admixed populations from UK Biobank. You can see here just in some examples here, each column in these plots is an individual, and you can see that each individual has varying proportion of African and European ancestry. And the advantage of studying admixed populations is that we have somewhat better control of environmental variation. So one of the difficulties in studying sort of cross-population genetic effects is that it's very difficult to separate out the effects of genetics and environment because they're often highly correlated. Now, in admixed populations, that's not to say there are no correlations between genetics and environment, but certainly we think those effects are smaller. So we think this is we said we have better control of environment, although I won't say that we have perfect control of environment. So what Barbara did was to go and compute polygenic scores in these individuals based on European GWAS, so in particular UK Biobank, and then to ask how the predictive power of those polygenic scores varies with ancestry. So what we've done here is we've actually split up each of these cohorts into sort of equally sized sub-cohorts, and then the x-axis shows you the proportion of European ancestry in each of these sub-cohorts. The y-axis shows you the partial R-squared, so the proportion of variance explained in that cohort. And what you can see is that there's essentially a linear relationship. So we fitted this grey line regression line to the admixed populations. Actually, this cohort, which is 100% European ancestry, is not included in the regression, but we have you can see that it actually predicts very well exactly where that cohort should be. So that really tells us that there's a direct relationship between ancestry and the predictive power of the polygenic score. In this case, in if you have 100% European ancestry, we explain something like 15% of the phenotypic variance. But if you have 0%, European ancestry, then we only explain something like 1 or 2% of the variance. So that's pretty alarming. And of course, the next question we want to ask is why that should be the case. So let's think about what we've shown. So we have two different ancestries. In one ancestry, in this case, this would be European ancestry, we have an R-squared of C%. In the second case, ancestry 2, in this case, for the sake of simplicity, let's say the R-squared is 0%. It's much lower than C. And what we've shown is that in an admixed individual, the R-squared, the predictive power, is roughly proportional to the ancestry. So if you have proportion of ancestry 1 x, your predictive power is roughly proportional to that. So then what happens if we then look at just the ancestry 1 regions? Okay, so we're going to mask out all the ancestry 2 regions and only look at the regions of ancestry 1. So what do we expect the R² to be in this case? Well, there's two really extreme possibilities. So in one model, all the predictive power comes from the ancestry 1 regions. And in that case, when we just look at the ancestry 1 regions, we should see the same that we see in the admixed individuals. So in that case, the predictive power should be equal to x, should be linearly proportional. But another model is that actually the predictive power still comes from the whole genome, but it's just proportional to x. Now, if that's the case, we actually expect to see a quadratic relationship between ancestry and R². You can think of it, if we add a little bit more ancestry, if we increase x a little bit, we're looking at a little bit more of the genome, but we're also increasing the overall level of predictive power, so we actually sort of double count those effects and we get this quadratic effect. And so what we actually see is that we can actually exclude model 1. Okay? And likely the true answer is somewhat intermediate, but certainly it's closer to what we would see with model 2. So another way to think about these two models is that model 1 is the case where what's important for determining the effect is actually the local ancestry, and model 2 is where what's important for determining the effect is actually global ancestry of each individual. And it seems like, at least from our data, at least in this particular phenotype, that model 2 is the one that's most important. And that's quite surprising, at least to me. It was surprising when we started this analysis. I was expecting local ancestry to be really the important factor, but actually it seems that that's not the case, and global ancestry is actually what's determining the predictive power here. And that's actually borne out by a follow-up analysis where we asked how we could actually combine polygenic scores. So we have here I'm showing you the predictive power in an African American ancestry cohort of different GWAS. So we have the GWAS from UK Biobank, and we have the GWAS from a diverse cohort, which is actually largely African American. And you can see that those two actually have similar predictive power, even though the diverse GWAS is much smaller, but it has similar predictive power because it's closer to the ancestry of the test population. And the question is, how could we combine these two to try and improve prediction in admixed population? And what we find is that actually if we use local ancestry information, so you use the European effect size in regions of European ancestry and vice versa, that actually doesn't do very well. In fact, that does worse than using either of the GWAS on their own. What actually does do well is combining them weighted by the global ancestry of each individual. Okay? Or similarly, just constructing a linear combination that we train weights for in another cohort. So again, that suggests that actually what's important here is not local ancestry, but actually really global ancestry. So again, that's kind of surprising. How should we think about this in the context of what it tells us about genetic architecture? Well, I would argue that this is actually an expected consequence of the omnigenic model proposed by Jonathan Pritchard and colleagues. So what does the omnigenic model say? The omnigenic model says that there are a relatively small number of genes that they call core genes that have a direct effect on the phenotype. But actually, most of the effects that we detect in GWAS occur in what they call peripheral genes, which affect the phenotype indirectly through this network of trans interactions. You can think of this as a gene expression network. You can make the same argument about environmental effects as well. So then the effect of perturbing any single gene, so the effect of a GWAS variant, is actually filtered through this network of complex network of other genes. So now when we go to another population, the values in the nodes of those networks change just because of genetic drift. Okay, so gene expression levels fluctuate randomly as we move to another population, and perhaps the topology of the network even changes as well. And what that means is that the marginal effect of any particular variant on the phenotype is likely to change as well. So I would argue that under the omnigenic model, we actually expect effect sizes to be different across populations. This is something that I think we and others will be able to test directly in the next few years as more diverse data sets come online. So that was all I wanted to say. I'd like to thank particularly Barbara, Arslan and Sam, postdocs in the lab who did the work I've presented here today. I'd also like to thank our funding sources, and I'd like to thank you for your attention. Thank you very much.